Hi, I'm Breyinn Loftin, and I will be discussing my research on enzyme-assisted peptide self-assemblies that trigger cellular adhesion in a high-density hydrogel. And so peptide self-assemblies have been recognized as an important component in responsive hydrogel materials that have applications in tissue engineering and regenerative medicine. And so this study focuses on the influence of the hydrogel matrix on the self-assembly behavior of the peptides, as well as their interaction with cells in order to further develop materials for these types of applications. And so we began by first developing a host gel, and this network is PEG-based, and it uses oxime click chemistry in order to form this very densely cross-linked polymeric network known as a hydrogel. And then for hybrid networks formation, the enzyme alkaline phosphatase is embedded within the host gel, and then upon diffusion of the peptide solution, the peptide is transformed into a hydrogelator that can then self-assemble within the host gel to form this intercalated network. And this network is confirmed using fluorescence emission spectroscopy and circular dichroism spectroscopy. And then we visualize the network using confocal laser microscopy, where we see that the self-adhesion net or the self-assembly network is distributed homogeneously throughout the host gel. And for the mechanical properties of first the host gel alone, we found that it does possess properties that are required for biological applications in regenerative medicine. And then once the peptide is allowed to self-assemble within the host gel, the mechanical properties don't change, which means that the mechanical stability of the network is not affected by the self-assembly process. And for the cellular adhesion properties of the network, we first examine the host gel, where we see little to no cellular activity. And then once the cells are introduced into the hybrid network, we begin to see cellular adhesion behavior and cellular activity. So this means that the cellular activity is promoted by the assembly of these peptides. And this feature can be further enhanced when we introduce the well-known cellular adhesion motif, RGD, into the system, because it also participates in the self-assembly process. It further enhances the cellular activity and cellular adhesion behavior of the cells. And so to conclude this work, we found that one, the mechanical stability of the network is provided solely by the host gel, and the cellular activity or cellular adhesion properties of the network is governed exclusively by the self-assembly of the peptides within the host gel. And this type of research opens the possibility to develop more diverse and advanced hybrid materials that can control cellular activity using a self-assembled network that is tailored to an environment provided by the host gel.